The overall goal of the following experiment is to generate large numbers of uniform, size-controlled tumor spheroids. This is achieved by first preparing the microwell plates to cluster the cells to be formed into spheroids. As a second step, a culture of the desired cell type is harvested, which provides the cell suspension that determines the size and composition of the spheroids. The cell suspension is then centrifuged into the microwells and incubated in order to allow spheroids to form. After incubation, the resulting spheroids are highly uniform and may be collected or cultured in the microwells in which they were formed.
The main advantage of this technique over existing methods is that very large numbers of uniform aggregates can be made simultaneously.
Individuals new to this method may find their spheroids vary in size. It is important to ensure that the cell suspension is evenly distributed across the plate and that the swinging plate carriers in the centrifuge move freely so that the force is perpendicular to the surface.
To begin, prepare a sterile Microwell 400 plate by adding 0.5 milliliters of surfactant-containing rinsing solution to each well. Using surfactant ensures that the cells do not interact with the microwell surface. To avoid trapping air bubbles in the microwells, add liquid at one side and allow it to spread across.